Welcome to our exploration of fluid, electrolyte, and acid-base balance in human anatomy and physiology. This chapter is fundamental for anyone studying health sciences, as it connects directly to interpreting patient lab results and understanding how the body maintains internal stability. We'll cover the basics of body fluid compartments, the movement of water and solutes, and how the body regulates these processes. By the end, you'll have a clear grasp of why these balances matter and how disruptions can lead to clinical issues. Let's dive in and see how these systems work together to keep us healthy. Understanding fluid, electrolyte, and acid-base balance is crucial for interpreting lab tests and patient results. These balances affect everything from hydration to nerve function and muscle contraction. When you know how to evaluate these values, you can spot imbalances early and help guide treatment decisions. Whether you're a student or a healthcare professional, mastering these concepts will improve your ability to care for patients and understand their physiology. Let's see why these balances are so central to health. Human body is composed of various fluid compartments. Infants have the highest water content, about 73% or more due to low body fat and bone mass. Adult males average around 60% water, while adult females have about 50%, reflecting higher fat content and less skeletal muscle. Adipose tissue is the least hydrated, and total body water in adults averages about 40 liters. As we age, water content declines to roughly 45%. These differences impact how fluids are distributed and how the body responds to dehydration or overhydration. Water is the universal solvent in the body, dissolving a wide range of substances called solutes. Solutes are classified as non-electrolytes and electrolytes. Non-electrolytes such as glucose, lipids, creatinine, and urea do not dissociate in water and are mostly organic molecules. Electrolytes, on the other hand, dissociate into ions and conduct electrical currents. This distinction is important because electrolytes have greater osmotic power, meaning they can cause fluid shifts between compartments more effectively than non-electrolytes. Electrolytes dissociate into ions in water, such as sodium chloride NaCl splitting in Na+ and Cl. These ions are crucial for conducting electrical currents and maintaining osmotic balance. Electrolyte concentrations are measured in milliequivalents per liter (mx/L), which reflect the electrical charges per liter of solution. For single-charged ions like sodium, 1 mEq equals 1 mOsm. For bivalent ions like calcium, 1 mEq equals 12 mOsm. This measurement helps us understand how electrolytes influence fluid movement and electrical activity in the body. Extracellular fluid (ECF) and intracellular fluid (ICF) have distinct compositions. ECF contains similar electrolyte contents, with plasma having higher protein and lower chloride levels. The major cation in ECF is sodium, while chloride is the major anion. In ICF, potassium is the main cation and HPO4 is the major anion. ICF also contains more soluble proteins than plasma. These differences are essential for maintaining cell function and overall fluid balance. Electrolytes are the most abundant solutes in body fluids, but the bulk of dissolved solutes consists of proteins, phospholipids, cholesterol, and triglycerides. These make up 90% of plasma solutes, 60% in interstitial fluid, and 97% in intracellular fluid. The distribution of these solutes is key to understanding how the body maintains its internal environment and responds to changes in fluid or electrolyte level. Fluid movement among compartments is regulated by osmotic and hydrostatic pressures. Water moves freely along osmotic gradients, and all body fluid osmolality is nearly equal. Changes in solute concentration in any compartment lead to net water flow. For example, increased ECF osmolality causes water to leave cells, while decreased ECF osmolality leads to water entering cells. These mechanisms ensure that cells and tissues remain properly hydrated and functional. Exchanges between plasma and interstitial fluid occur across capillary walls. Fluid leaks from the arteriolar end of capillaries and is reabsorbed at the venule end. Lymphatics pick up remaining fluid and return it to the blood. Exchanges between interstitial fluid and intracellular fluid happen across cell membranes, with two-way osmotic flow of water and selective movement of ions. Nutrients, wastes, and gases move in one direction, ensuring proper cellular function. Water balance and extracellular fluid osmolality are tightly regulated. Osmolality is maintained around 280-300 mOsm. A rise in osmolality stimulates thirst and causes antidiuretic hormone ADH release, while a decrease inhibits both. These mechanisms ensure that the body maintains proper hydration and electrolyte levels, preventing imbalances that can affect health. Thirst mechanism is the driving force for water intake, governed by the hypothalamic thirst center. Osmoreceptors in the hypothalamus detect changes in extracellular fluid osmolality and are activated by increased plasma osmolality, dry mouth, decreased blood volume or pressure, and angiotensin II or baroreceptor input. Drinking water inhibits the thirst center, with feedback signals like relief of dry mouth and activation of stomach and intestinal stretch receptors. Obligatory water losses include insensible water loss from lungs or skin, sensible water loss from urine, sweat, and feces. The volume of urine excreted and solute concentration depend on fluid intake, diet, and other avenues of water loss. These mechanisms help the body maintain water balance and prevent dehydration or overhydration. Antidiuretic hormone ADH plays a key role in regulating water output. When ADH levels rise, water reabsorption increases, reducing urine output. When ADH is inhibited, water is not reabsorbed, leading to increased urine output. This hormone helps maintain fluid balance and responds to changes in osmolality and blood volume. Dehydration is a principal abnormality of water balance, characterized by extracellular fluid water loss. Signs and symptoms include a cottony oral mucosa, thirst, dry flushed skin, and oliguria. Severe dehydration can lead to weight loss, fever, mental confusion, hypovolemic shock, and loss of electrolytes. Recognizing these symptoms is vital for timely intervention. Hypotonic hydration, also known as water intoxication, occurs with renal insufficiency or rapid excess water ingestion. Extracellular fluid osmolality decreases, causing hyponatremia and net osmosis of water into tissue cells, resulting in cell swelling. Symptoms include severe metabolic disturbances, nausea, vomiting, muscular cramping, cerebral edema, and possible death. Treatment involves hypertonic saline. Edema is the atypical accumulation of interstitial fluid, resulting in tissue swelling but not cell swelling. Only the volume of interstitial fluid is increased. Edema can impair tissue function by increasing the distance for diffusion of oxygen and nutrients from blood into cells. It may be caused by increased fluid flow out of blood or decreased return of fluid to blood. Electrolyte balance usually refers to salt balance, though electrolytes also include acids, bases, and some proteins. Salts enter the body through ingestion and metabolism and are lost via perspiration, feces, urine, and vomit. Maintaining electrolyte balance is essential for nerve function, muscle contraction, and overall cellular health. Electrolyte imbalances can have various causes and consequences. For example, deficiencies or excesses in sodium, potassium, calcium, or chloride can lead to symptoms ranging from muscle weakness to cardiac arrhythmias. Understanding these imbalances helps in diagnosing and treating conditions related to fluid and electrolyte disturbances. Further causes and consequences of electrolyte imbalances include conditions like Addison's disease, where too little aldosterone is produced, and pica, a disorder where individuals eat non-food substances due to mineral deficiencies. Recognizing these clinical signs is important for identifying underlying electrolyte disturbances. Sodium is the most abundant cation in extracellular fluid and is critical for fluid and electrolyte balance. Sodium salts contribute significantly to the osmolality of ECF, controlling ECF volume and water distribution. Changes in sodium levels affect plasma volume, blood pressure, and fluid volumes in ECF and interstitial fluid. Sodium movement is tightly regulated, and its transport is linked to renal acid-base control mechanism. Sodium concentration determines the osmolality of ECF and influences the excitability of neurons and muscles. It remains stable due to water shifts between compartments. Sodium content, on the other hand, determines ECF volume and blood pressure. Understanding the difference between concentration and content is important for interpreting lab results and managing fluid balance. There are no known receptors that monitor sodium levels directly in body fluids. Sodium-water balance is linked to blood pressure and blood volume control mechanisms. Changes in blood pressure or volume trigger neural and hormonal controls to regulate sodium content, ensuring proper fluid and electrolyte balance. Several hormones influence sodium balance. Estrogens increase sodium chloride reabsorption, leading to water retention during menstrual cycles and pregnancy. Progesterone decreases sodium reabsorption, promoting sodium and water loss. Glucocorticoids also increase sodium reabsorption and can promote edema. These hormonal effects are important for understanding fluid shifts in different physiological states. Cardiovascular baroreceptors alert the brain to increases in blood volume and pressure. When activated, sympathetic nervous system impulses to the kidneys decline, causing afferent arterioles to dilate, increasing glomerular filtration rate (GFR), and promoting increased sodium and water output. This reduces blood volume and pressure, helping maintain homeostasis. Potassium is vital for resting membrane potential in neurons and muscle cells, especially cardiac muscle. Hyperkalemia, high potassium, causes depolarization and reduced excitability, while hypokalemia, low potassium, causes hyperpolarization and non-responsiveness. Disruptions in potassium levels can interfere with heart electrical conduction, leading to sudden death. Potassium also plays a role in the body's buffer system, with levels rising in acidosis and falling in alkalosis. Potassium balance is controlled in the distal convoluted tubule DCT and collecting duct by regulating the amount secreted into filtrate. High potassium content in extracellular fluid favors secretion, while low levels reduce secretion. Type intercalated cells can reabsorb potassium left in filtrate. The kidneys have limited ability to retain potassium, so most is lost in urine, making dietary intake important. Aldosterone stimulates potassium secretion and sodium reabsorption by principal cells. Adrenal cortical cells are directly sensitive to potassium content in extracellular fluid. Increased potassium in the adrenal cortex causes aldosterone release, which increases potassium secretion. Abnormalities in aldosterone can cause swift and lethal hyperkalemia or severe hypokalemia, leading to paralysis. Calcium in extracellular fluid is important for blood clotting, cell membrane permeability, secretory activities, and neuromuscular excitability. Hypocalcemia increases neuromuscular excitability and can lead to muscle tetany, while hypercalcemia inhibits neurons and muscle cells and may cause heart arrhythmias. Phosphate regulation is closely linked to calcium, with phosphate acting as a buffer in urine and intracellular fluid. Chloride is the major anion accompanying sodium in extracellular fluid, helping maintain osmotic pressure of blood. 99% of chloride is reabsorbed under normal pH, passively following sodium in the proximal convoluted tubule and coupled to active transport in other segments. Other anions, like sulfates and nitrates, have transport maximums and excesses are excreted in urine. pH affects all functional proteins and biochemical reactions, so it is closely regulated by the body. Normal pH of arterial blood is 7.45, venous blood and interstitial fluid is 7.35, and intracellular fluid is 7. Alkalosis is arterial pH above 7.45 and acidosis is below 7.35. Maintaining acid-base balance is essential for health and cellular function. Small amounts of acidic substances enter the body in food, but most hydrogen ions are produced as byproducts of metabolism. Phosphorus-containing protein breakdown releases phosphoric acid, lactic acid comes from anaerobic respiration, fatty acids and ketone bodies from fat metabolism, and hydrogen ions are liberated when CO2 is converted to bicarbonate in blood. Concentration of hydrogen ions is regulated by three mechanisms: chemical buffer systems, rapid first line of defense, brain stem respiratory centers acts within 1-3 minutes, and renal mechanisms most potent but require hours to days. These systems work together to maintain pH within a narrow range. Chemical buffers resist pH changes when strong acids or bases are added. Strong acids dissociate completely and can dramatically affect pH, while weak acids dissociate partially and are efficient at preventing pH changes. Strong bases dissociate easily and tie up hydrogen ions quickly, while weak bases accept hydrogen ions more slowly. There are three major buffering systems: bicarbonate buffer system, phosphate buffer system, and protein buffer system. Each acts to resist pH changes by binding or releasing hydrogen ions as needed. These systems are distributed throughout the body and play key roles in maintaining acid-base balance. Bicarbonate buffer system consists of a mixture of carbonic acid HCO3 and salts of bicarbonate HCO3, such as sodium bicarbonate. It buffers both intracellular and extracellular fluid but is the only important extracellular fluid buffer. When strong acids or bases are added, the system ties up hydrogen ions or releases them to maintain pH. Phosphate buffer system works similarly to the bicarbonate system, using sodium salts of dihydrogen phosphate HPO4 and monohydrogen phosphate HPO42. It is effective in urine and intracellular fluid where phosphate concentrations are high. This system helps tie up hydrogen ions from strong acids and convert strong bases to weak bases. Intracellular proteins are the most plentiful and powerful buffers, with plasma proteins also playing a role. Protein molecules are amphoteric, meaning they can function as both weak acids and weak bases. Hemoglobin is an important intracellular buffer, helping maintain pH in red blood cells. A quick summary of chemical buffer systems shows how bicarbonate, phosphate, and protein buffers work together to maintain pH. Each system has its own strengths and locations in the body, ensuring that pH changes are resisted and balanced efficiently. Respiratory system acts as a physiological buffer, eliminating carbon dioxide, which is an acid. A reversible equilibrium exists in blood, allowing CO2 and water to form carbonic acid, which dissociates into hydrogen ions and bicarbonate. During CO2 unloading, the reaction shifts to the left, incorporating hydrogen ions into water. During CO2 loading, the reaction shifts to the right, buffering hydrogen ions with proteins. Blood PCO2 rises, hypercapnia. Medullary chemoreceptors are activated, increasing respiratory rate and depth. Rising plasma hydrogen ions, acidosis, also activate peripheral chemoreceptors, increasing respiratory rate and depth. Both responses remove more CO2 from the blood, reducing hydrogen ion concentration. Alkalosis depresses the respiratory center, decreasing respiratory rate and depth, causing hydrogen ion concentration to increase. Chemical buffers cannot eliminate excess acids or bases from the body. Lungs eliminate volatile carbonic acid by removing CO2, while kidneys eliminate non-volatile acids produced by metabolism, such as phosphoric, uric, lactic acids, and ketones. Kidneys also regulate blood levels of alkaline substances by adjusting the amount of bicarbonate in the blood. Maintain alkaline reserve, kidneys must replenish reabsorbed bicarbonate. Tubule cells are impermeable to bicarbonate but permeable to CO2. Bicarbonate can gain entry back into the body by being converted to CO2, which can then be converted back to bicarbonate inside the cell. This mechanism is coupled to hydrogen ion secretion, with every hydrogen ion secreted leading to bicarbonate reabsorption. Metabolism of food generates new hydrogen ions, which must be balanced by generating new bicarbonate. Secreted hydrogen ions used to reclaim filtered bicarbonate are incorporated into water molecules, which can be reabsorbed. Two mechanisms in the proximal convoluted tubule and type intercalated cells generate new bicarbonate by excreting buffered hydrogen ions or ammonium ions. Most important urine buffer is the phosphate buffer system. Intercalated cells actively secrete hydrogen ions into urine, which are buffered by phosphates and excreted. New bicarbonate is generated in the process and moves into interstitial space, then passively into peritubular capillary blood, replenishing the alkaline reserve. All acid-base imbalances are classed as either respiratory or metabolic. Respiratory acidosis and alkalosis are caused by failure of the respiratory system to perform its pH balancing role, with blood PCO2 as the key indicator. Metabolic acidosis and alkalosis are indicated by abnormal bicarbonate levels. Understanding these imbalances is crucial for diagnosing and treating acid-base disorders.